Good morning and good afternoon, everyone. Welcome to today's broadcast, Massively Parallel Genome Engineering for Rapid Target Discovery in Microbes, being presented today by Dr. Stephen Federowicz, a Senior Scientist of Computational Biology at Inscripta. My name is Andrea, I'm with LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is brought to you by LabRoots and sponsored by Inscripta. For more information on our sponsor, please click on the sponsor logo on the left side of your screen, or you can visit inscripta.com. I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you like at any time during the presentation. To do so, simply type them into the Ask a Question box and click on the Send button. We will be answering as many questions as we have time for at the end of the presentation. You can also use this Ask a Question box to report any trouble you might have hearing the presentation, or you can click on the Support tab found at the top right of the presentation window. Also, please notice you can share this webinar on your personal social media. Just click on the Social Sharing tab to let your friends and colleagues know about today's live event. Now I'd like to introduce today's presenter. Dr. Stephen Federowicz is a bioinformatician and systems biologist with over 5 years of industrial experience in next-gen sequencing analysis, network biology, algorithm development, and machine learning. He is passionate about developing genome-scale understandings of organism function with high-throughput experimentation and computational techniques. Dr. Federowicz is a Senior Computational Scientist in Inscriptas Applications Division, where he is responsible for the analysis and interpretation of the complex datasets generated from the Onyx Digital Genome Engineering Platform. And with that, Dr. Federowicz, I'll turn it over to you to begin.
All right, thank you so much for being here with me today. My name is Steve Federowicz. I'm a Senior Data Scientist in the Applications Group at Inscripta. I'm very excited to talk with you today about massively parallel genome engineering for rapid target discovery in microbes. So to jump right in, onto the outline here, first thing we're going to do is give an overview of the Onyx platform. We're going to start with a brief background on some of the core technology, and then we're going to talk a little bit more about the specifications and capability. From there, we're going to jump into a more expansive data discussion around genome-scale target discovery in E. coli. We're going to first talk about how we generate over 25,000 precise edits across 7 genome-scale libraries. We're then going to talk about how we subject these 25,000 edits under pool growth collections across 11 industrially relevant stress conditions. We're then going to spend most of our time talking about the characterization of these over 800,000 data points that come from these relatively simple experiments. And then finally, we're going to talk about the genotype-to-phenotype map of response inhibitory compounds that result from this data. And specifically, we're going to primarily focus on the toxic inhibitor compound furfural. We'll also spend a little bit of time talking about vanillin. And importantly, in this section, what we'll do is we'll spend a good amount of time really confirming the existing literature. You'll see how in these experiments, decades of work can be recapitulated in just a few months beyond this platform. And then finally, we'll talk about, after confirming the existing literature, we'll talk about the discovery of hundreds of novel targets that exist in these coordinated response patterns and can be readily used for strain improvement. So let me jump right in, again, for a brief overview of the Onyx platform so you understand kind of how the data is generated and what we're talking about. So the background of Inscripta and Onyx really goes back to this publication in 2016 in Nature Biosec. Our co-founder, Andrew Garce, showed that you could link a guide RNA with a donor molecule. And doing that in mass would allow you to do pool multiplex recombination to generate thousands of individual strain variants at once. We were obviously very excited about this. At the time of Nature Biosec was renamed to Inscripta. And it was great to see that throughout the field, and if you're coming from the academic side, you're more familiar with some of the additional publications. In the spring of 2018, there was a series of wonderful publications, really using this basic technology, make some improvements here and there. But for the most part, it was really exciting for us to see the community interest in generating these types of libraries. It's really our goal at Inscripta to make this easy for anyone. So with that, I had a little bit there. So with that, the key aspect of Inscripta technology is that we believe that biologists need high-throughput tools to make diverse edits genome-wide. And so this graph is what we like to think about for motivating the work. On the y-axis, we have a variety of edit types. And on the x-axis, you have the number of genomic locations that can be accessed. So in this left-hand quadrant here, across maybe 10 or 100 genomic locations, you can easily do knockouts and knock-ins. For protein engineering or path engineering, you can substitute in 10 or 100 different ORs, in plate-based workflows. And then also you can do NNK type saturation and genesis and achieve very high number of edits at a very low number of loci. Similarly, if you're looking at the lower right-hand quadrant, you can see in cases where you have a low variety of edit types, so genome-wide base editing, where you're doing SNPs or genome-wide knockouts, especially in yeast or classical genesis, you can access a large number of genomic locations, but you're going to be limited by the number of edits, the number of different types of edits you can make at those large number of locations. The power of the Onyx platform is in this upper right-hand quadrant, where at a large number of genomic locations, we can make a large variety of edit types. The Onyx platform, as I'll also introduce, is an all-in-one system, software, consumables, and instrument. It's really a push button to enable you to make any edits that you can think of. So current technology is something that happens generally in a microtiter plate. So our familiar 96-well plate, in each well of that plate, you might put the oligo guide RNA for cut, and simultaneously put in the donor or oligonucleotide that would be conferring the edit that you wanted. And this is excellent, and you can achieve high editing rates, but again, you're only getting about 96 edits at once. Really, the core of the Inscripta technology is to, instead of have a plate-based partition, we're going to move that partition to the individual cell. And so, again, that core technology that I showed, really paper from Garst et al., the key innovation is to synthesize the guide RNA and the donor oligonucleotide on an oligonucleotide array that would normally be used for hide capture or for other pull-down methods. By doing this, you can synthesize hundreds, up to 100,000 of these. And so what Inscripta does is, again, we synthesize these in mass, cut and paste, and we also add a trackable barcode that can be sequenced to know what cells were edited and what's being enriched and depleted in your population. The data that comes from this barcode is the primary data source we'll talk about in today's presentation. So what are the specs of the system? How does it work? Again, real quick, this system currently is for E. coli and yeast. We have standard strains that we support, but also any custom strain. Obviously, with strain engineers in the audience, when we talked about rapidly improving tolerance, in the data we'll present today, and you'd likely be doing that tolerance on a custom strain that maybe already is producing your molecule of interest with maybe a large number of heterologous genes or other edits already in there. The current platform will be one edit per cell. The edit types are insertions, deletions, or swaps, up to 60 base pairs. So that's long enough, as you'll see today, for promoter insertions in E. coli. And you can even do, of course, you can just do individual SNPs if you like. The number of designs currently supported on the platform is 100 to 100,000 or 100 to 10,000 per library. And one of the key metrics that we'll talk about coming up here, and really one of the real reasons to build this integrated microfluidic system and all this software is the percent edited cell metric. And that's saying, of the pool of cells that comes off of the instrument, how many of them have the exact edit that you want? As we'll talk about in a minute, it turns out that that's actually hard to optimize for, and that's one of the key values of the Onyx Platform. We'll also have design coverage, so of the 10,000 you design, making sure that you get good representation and then the turnaround. All right, so some examples of demonstrated types. So again, I mentioned up to 60 base pairs. So one thing you might want to do is this promoter RBS insertion. As we'll talk about in a minute, you can do things like IGEM promoter ladders or other heterologous elements that will drive expression, make defined perturbations to your system. You might want to do terminator swaps. We've demonstrated this in other presentations. Doing these in yeast to tune expression up or down. So hitting with 5' and 3' PRs. Knockouts or triple stop swap codons. So in this case, it can be a little more robust. The normal edit type from CRISPR, right, is making a cut and then having it be paired with non-homologous end joining. Here what we're saying is we want to make a very precise stop codon at a precise place and ensure that truncated proteins other things don't form. And then finally, a very interesting one is saturation genesis. Any position across a protein, you could introduce alt codons to mutate that protein to every other amino acid. All right, and so 10,000 custom designs. You think about each of these little dots being a different design that you would make. And what comes off the machine is those 10,000 designs are made across a population of cells. The result is millions of precisely engineered single cells that are going to generate thousands of distinct phenotypes. And so when we're making an edit, what do we mean by that? Inscripta is very particular and very proud that when we say an edit, what we mean is a complete and intended edit. So that's best illustrated here. So in the first row is the wild type sequence, where this is the target. So say you're trying the wild type sequence is a PAC, you're trying to change it to ACT. Inscripta will also generally make auxiliary edits, altering the PAM or still base region to have immunizing effects, so there's not constant recutting. And what often happens is that this is what you want. You want the complete and intended. So you designed to change this codon, and you wanted to alter the PAM site. What often happens is that you get either complete with unintended or partial incomplete. So what often happens is that the auxiliary edit will go in to do immunizing to prevent recutting, but your designed edit does not go in. This is a common feature of the Onyx site chemistry, and something that we've been very careful to optimize for, and to be very clear that when we present editing data, we're only ever talking about complete and intended. The exact sequence that you designed, you wanted for the genome, is the sequence that we're providing. And again, like as you can see in this graphic on the right, if you look at all the chromosomes in yeast, we're talking about just one state. And this would be saying it was 90%, would mean that it wasn't that there was 90% cutting, but 90% of the cells are the exact edit. So to make that, to break that a little bit more, kind of the main measures of performance we're talking about would be edit efficiency, which would be generally talked about at a single isolate. So in the Onyx system, if you did look at a single isolate, our edit efficiency is greater than 95%, and that again includes that complete and intended exact thing you designed. To contrast that with the literature, oftentimes what you see in the literature is edit efficiency is reported as greater than 90%. Those often include NHEJ and HDR, and they don't report complete and intended. You can also get errors in the synthesis process, and that's something that we control for and correct for, but are would not have been treated for in those publications. The next most critical measure of performance is percent of edited cells. That I was talking about. And again, this applies not to a library. We're not talking about an isolate of one edit. We're talking about thousands of edits. The percent of edited cells contain those thousands of edits. A typical Onyx value would be greater than 50%, right? So if you have 100,000 cells, 50,000 of those cells are going to be your complete intended edits. Whereas in the literature, in the best case scenario, you might get around 5%. reported for the method. Finally, design to preserve. So if you're looking around the genome, all the different designs that might come up, we have optimized to ensure that on the Onyx platform, you're going to get greater than 99% in the literature coverages that are not reported or can be highly rare. But again, the data we'll be showing will be E. coli, but as we said, target discovery in microbes. Onyx looks on both E. coli and yeast, and so here's just a little bit of yeast performance data across a number of different library types: saturation mutagenesis, substitution of indels, transcription binding site swaps, mixed libraries. You can see that we achieve a very good percent of edited cells, percent of complete intended edits, 75% to 30%. And again, this would be if you had 100,000 cells, 750,000 of those cells would be the exact edits that you wanted in your amino acid saturation library. All right, so shifting and getting closer to the data now. I'm going to give you a background on the platform and what it is you understood the data I was talking about. So the general way we think about often our workflows is that a pool of edited cells come off the Onyx instrument. There's two main workflows. First workflow is a pooled workflow where you can do selections. Then you'll sequence those barcodes I talked about. And then from all that data, you'll do analysis and learn. You can learn new insights and move forward with your studies. The other workflow that I won't talk about today, I've talked about in the past and in other reference material on the Inscripta website, is to take those edited cells, put them back into isolates, and then do HTTP direct detection or other methods to directly test individual isolates. And there's a lot of powerful aspects of both approaches, complementary. And again, today we're going to talk about these pooled sequence approaches. All right, so jumping into the data here. So a problem, common problem in the transition from a petrochemical to a bio-based economy is having cost-effective feedstocks at scale. So many of you might be familiar with the problem, a very common problem with biomass like switchgrass or cellulose. You want to use cheap feedstock like that to ferment valuable products at scale, but oftentimes through that fluid acid hydrolysis or other methods to break down that biomass result in not only the fermentable carbon sources that you want your bugs to eat, but also a number of other potentially inhibitory or often has inhibitory compounds. And right, one thing that commonly happens is that you'll start fermenting some fluid acid hydrolysis. You'll have a low pH. To balance out that pH, you'll add some sodium hydroxide or the base. So now you have, subjected your bugs to inhibitory compounds, a pH problem, and now a salt problem. And so as you'll see in this study, we undertook a massive a campaign to determine all the targets that would respond genotypically to each of the genotypic challenges. And so it's obviously many decades of excellent work has resulted in the discovery of these pathways and mechanisms to overcome this inhibition. But again, there's a perennial need to determine the response to new compounds, especially in our new strains and new production conditions. And again, here today we're going to show how decades of work can be recapitulated in just a few months. And so the first step, let's design and generate some edits. Okay, and so to design our libraries here, we're going to start with the IGEM promoter ladder. Maybe familiar to many of you, and there's 20 separate promoters here, all derived from one common promoter from E. coli. And in our library, we're also going to introduce gene knockouts by introducing triple step. So the promoter ladder is great because it has a lot of relative strength. Unfortunately, it's not in order, but this promoter here, the 112, is kind of the native promoter. But then going throughout this, the 116, the 106, the 102, and the 100 all have different relative strengths. And these happen because of these minor modifications to -35 and -10 tadabox that enable these promoters to drive expression much higher in a constitutive manner. And so what we wanted to do was take these promoters, pick these promoter sequence, and ultimately we're not showing here the RDS as well, but we did a full 60-base pairs, and insert them in front of every gene on the genome. And then also insert these gene knockouts at position 5 and 15 of every position on the genome. And so what does that look like? So if you have the coli genome here, going around the familiar circle spot, the first 7,933 knockout edits, again, we made one knockout at position 5 and one at position 15 of every gene in the genome. So that's kind of what this looks like going around here. If we then add the J23112 promoter, the weakest promoter at 1x, we have another 3,676 edits, 116, 106, 101, 100, for a grand total of 25,402 genomic edits. And so hopefully that you get this view here can give you an appreciation of the scale of really editing every gene in the genome, the type of data that we're going to be able to get back. And again, right, if you do a cross-section and look at just one gene, at each gene, it would have all 5 of the different promoters in front of it, and each of the 2 different knockouts, and that would be in 7 distinct cells, each with one of those edits. All right, so after designing, it's time to take our engineered cells off the instrument. They've been designed, software, as we said, has been generated. Now we have our pool of edited cells. We're going to run our selection experiments. So what does that look like? So I mentioned earlier we're going to look at salt, pH, and biomass inhibitor tolerance. We have 25,000 edits now in 1, 2. We've pulled all these libraries together. We're going to focus first on furfural, vanillin, hydroxymethylfurfural, and citric acid. We're going to do biological triplicate growth selections for each condition. We'll do 48-hour cultivations of the harvest. We also have a control condition. It's not shown. Again, this is going to target most genes in E. coli, 2,676. Some weren't targeted due to design constraints, although many these are earlier reagents in development, so that number will go up over time. We have a 5x promoter ladder, the 2x knockout ladder, and again, it's 25,000 total designs. I talked a lot about the fraction of edited cells earlier on. These libraries, it's roughly only 18 to 25% for each of the promoters. Yeah, and again, so to give you a sense of scale, we ran selections across all of these. And again, for today's talk, we're going to mainly just focus just on the biomass inhibitors for all vanillin, HMF, and citric acid. All right, so we engineered the cells, made our selections. Now it's time to sequence. That looked like. So we talked about the setup, the Onyx chemistry. There's really a guide RNA for cutting, a donor DNA molecule for pasting that's going to confirm that edit that we want, and a trackable barcode. Again, these are multiplexed into all the cells. And what we end up sequencing is we're sequencing the barcodes. And so shown here again, to go back to the circular plot, the coli genome. What's plotted down the y-axis here is just the log2 of the raw count to come off with the sequencer from these barcodes. I'm showing here a control condition. And you can see that in the control condition, the knockouts, you see a dispersion of counts across the genome. So each one of these dots is an individual cell that has an individual edit in front of its in its gene, in this case knockouts. When we look at the weak promoters, now altering the expression of every gene in the genome in all these individual cells, the average strength promoter, strong promoter, stronger, and then strongest. And even now in these counts, as you click through a little bit, you can see especially going from weak to average, you start to see a general increase in the counts along the y-axis. As we dive in here to this data, we're going to talk about how to look at this differentially, how to treat it statistically, and how to quantify that. Okay. So after our sequencing, it's time to do some analysis and learning from our data. And so the question here is, how do you analyze 25,000 edits? So here we took a four-step process. The first thing we did was we wanted to assess the quality and biological reproducibility of the data. Second, we wanted to determine a method to look at differences in the data amongst conditions. How do we look at this comparatively? And then we want to look at genome-scale trends, enrichment and depletion. Are there any obvious things that pop out? And then finally, we want to do a functional assessment of the enriched, depleted genes, targets to confirm existing knowledge, see if we can reveal some new findings. So what comes off after you've done your barcode sequencing is this lovely data table. So in this case, right, we have 25,000 edits. So this table would have 25,000 rows. We might have it show each edit here, show each gene that the edit is made on. And then across each of our 3 biological replicates, we have our furfural, or vanillin, or syringic, and our 5-hydroxymethyl furfural is those 4 inhibitory compounds. And so in all, this is 831,000 data points. So how do we look at this? How do we deal with it? It looks a lot maybe like any other RNA-seq account data set. We do take some pages from that book and the other CRISPR literature to analyze the data. So if you plot a large correlation matrix, you can immediately see that there's a lot going on. The first thing I'll draw your attention to is the diagonals. So along the diagonals, you're seeing among furfural, among each of the 1, 2, 3 biological replicates, you're seeing the cross correlations. See this for each condition, 5-HMS, syringic, vanillin, and control. We're seeing, for example, in the control, very good biological reproducibility, greater than 0.94, 0.97 for controls. These are Pearson correlation. The experiments are also equally high. And across furfural, again, you're seeing greater than 0.95 correlations across complete biological replicates. And these are biological replicates of this pool of 25,000 edits rolling up in selection, and we're sequencing those barcode counts. So in some ways, it's kind of amazing to do with it. And so the next thing you can look at is the correlations amongst individual inhibitors and the no treatments for range of dispersion. So the main thing we're going to focus on here today is the control versus the furfural. We'll also look a little bit about control versus the vanillin. And that difference of correlations of 0.86 versus 0.9 is effectively the larger biological difference you're seeing happen given the larger perturbation that furfural is making. So another way to look at this data is to do a principal component analysis. So we do the principal component analysis among all of the data now, salt, pH ladder, pH gradient, and each of these 4 inhibitors. What we see is some really nice separation. So up at the top, you get the 3 furfural replicates coming together very cleanly, top of the second principal component. And what's really nice to see is along the first principal component, we see both the salt concentration gradient and the pH gradient essentially falling out along that component. Really great to see not only the replicates coming together, but some of the biological significance being shown clearly in the principal component. And so now, second step here, let's take a look at some of the comparative genome-wide responses. So now what we're going to be plotting on the y-axis is the log2 of the treatment count versus the control count. So we'll define that a little more going forward, but in general, you can think of this just like any fold change we do for an RNA-seq expression experiment. And so on the left-hand side here, this is going to be furfural over control, the control condition, which would be no inhibitor added, just glucose M9. And then over here would be vanillin over the control. So again, 1 milligram over the vanillin with base M9. And you can see a bit difference in the response. You can see that we're showing here first for the weak promoters across all the genes or on the genome. Things like this, where it's at this gene at an enrichment, fold enrichment of 2 is, what you would consider a hit, had a strong response by putting a weak promoter in front of that gene. Over here you have a massive hit, almost a 4-fold response from that, putting a weak promoter in front of that particular gene. But overall, it's a generally balanced pattern around the genome. Same thing with vanillin. What gets interesting is you start to go out, the average promoters, strong promoters, stronger promoters, or the strongest promoters. You start to see a little bit of the pattern of, especially even starting with the strong and stronger promoters, you're getting more large hits. Because again, we're constitutively driving expression higher, so you'd expect there could be, for genes that are conferring a disadvantage, when they're expressed even higher, they might confer an even larger fitness advantage. Or for genes down here, overexpression is even more detrimental to the cell under vanillin challenge. And in the spirit of biology always being interesting and always being learning something new, when we looked at the knockout, we immediately see the striking pattern, which shows up a little bit in the promoters, where, if you're looking here along the, this entire megabase of E. coli genome, you see this dramatic enrichment. And, looking a little bit more, sitting by the origin of replication, we know that E. coli genome has high copy number or higher copy number, but have multiple replication forks in log growth. We don't know why this is asymmetric. A lot of the ribosomal genes sit here. But it's just one interesting thing to point out that you use a system like this and look at new biology, all kinds of interesting things pop out. All right. So after looking at the genome scale, we're going to dive in a little bit and look at some of the specific mechanisms and focus in particular on furfural. So there's been this excellent body of work on the characterization of furfural tolerance from Ingram et al. And really the key mechanism of furfural tolerance is NADP starvation. So what happens to detoxify furfural alcohol, this by QHD active reductase basically reduces furfural, and it's specific to NADPH. Since your cell has a more limited amount of NADPH, this actually causes starvation, and the cells will lower growth. So in excellent work over a long period of time, Ingram et al. Have characterized this in detail. They've knocked out these genes YQHD, they've overexpressed PNTA, knocked out YQHD. There's a number of really great detailed studies to elucidate this mechanism. And also showing, that this transhydrogenase here, in particular, it's like a larger fold NAD than NADP. Overexpression of transhydrogenase, which can occur between the two, can rescue the furfural effect. Okay, so we look at furfural response overall. We're going to look at one gene now. So we're going to look at that PNTA gene, PNTA transhydrogenase, that was going to help to rescue this furfural growth defect by making more NADP. So we look at this one gene, let's point out here, this one dot here, where it's increase in expression, increase in enrichment. You can see that in furfural, the barcode counts coming from the NGS are high, and the control they're low. And really what we're going to be talking about for the rest of the data here is really these slopes and biological responses. It's really just laws of fold change between the control and the condition. And what's interesting is we can look at these across the library of edits of all of the 7 different edit types we made. So each of the knockouts, position 5, the knockout at position 15, and then the increasing promoter strength ladder. And again, each case is furfural versus the control. And you're seeing, even just looking at the raw counts, there's maybe a mild effect from the knockouts, a similar effect from the weak promoter. In many cases, a very weak promoter might be effectively biologically equivalent to knocking out the gene. And then under increasing promoter strength, you see that you start to see this effect. So there's the average promoter, you start to see that there's enrichment, that the edit on this gene is conferring furfural tolerance. And you sort of see this Goldilocks effect where the medium strength promoter actually has the largest effect. It seems to help the most. And then maybe at these stronger promoters, you're actually expressing too much of this gene. So even though it helps, the overexpression starts to cause a fitness defect. And if we zoom out now, right, we were looking at just one gene there. When we zoom out and look at all of our 25,000 responses, this is what it looks like. So we've highlighted here, the black dots are the PNTA gene. So that was the slopes we were just looking at. But you can see that that one detailed profile of that one gene is one of the 25,000 that you can get. You're going to see the broad response patterns overall. And so looking at this differentially now, and so we do the differential analysis, we use DC2 in this case, for count data to get statistics. If we look at the implanted Dalton plot, what we're seeing now is the mean cassette count plotted on the x-axis, with log twofold change plotted on the y-axis. And then we've just highlighted less than 1 meter than -3rd, significantly enriched or depleted edits. And so on this axis, really, anything that's enriched in the population relative to the control is considered making the cell more resistant to furfural. Anything that's depleted would mean that we made an edit that made that cell less resistant. And so what we've highlighted here in these red dots are all of the previously reported genes that are known. So some of the genes we were just talking about, the first was PNTA. If we look over here, we can see that indeed this transhydrogenase that was heavily validated, through decades of work to show that it does help in increasing furfural tolerance, indeed shows up at some of our biggest hits. We're seeing multiple cases of it because, again, multiple different promoters of different constitutive strength all drove the same response into being more resistant. STHA was on the diagram. It's another for the other key transhydrogenase in E. coli, and in this case, knocking it out doesn't help, which is expected. The second key gene is YQ8D, which has been validated that the knockout helps resistance by decreasing the depletion of NADPH. Again, shows up as one of our biggest hits. And then a few other genes that are also octoreductases and known in the furfural response also show up predictably. And so the exciting thing for us is that there's over almost 3,800 edits are identified to kind of confer resistance or depletion across the genome-wide library. And the key point is that these previously validated genes clearly show up, but there's many other edits on many other gene targets that seem to also confer great resistance to furfural. And so if you're a strain engineer and you are trying to rapidly determine all the genetic changes that can get you to an optimum production level, and you have optimum scale-up properties, a simple study like this can elucidate hundreds of targets for you to go after. So we just looked at what genes went up and down. So another way to look at this is to look in clustering. So a common way to look at clustering data in the single-cell world is to use UMAP dimensionality reduction followed by HDB scan clustering. And by doing this across a matrix of 990 of our genes of interest that are highly enriched or depleted across all 6 genotypes, looking at that matrix, we get these 4 very distinct clusters that emerge. And if we take a second and we dive in and look at each of these clusters, we can see that for this first cluster, for example, where we have 222 genes, each dot is an individual gene, we can look at a box plot of the response of all those 222 genes. And a striking pattern emerges where for these 222 genes, we see a pattern where the knockout always generally depletes, meaning it's not helpful for furfural tolerance, whereas each of the promoters, in an increasing manner, seem to help confer additional furfural tolerance. So we call these tolerance-conferring cluster models. In our familiar genes, we're just talking about our transhydrogenase, KTA, there's other octoreductases implicated in the furfural response, show up in this cluster as genes where if you don't want to knock it out, you want to overexpress these genes to help your strain. If we look at some, functional enrichment of this, we see some really interesting things. This acyl transferase activity related to 7-4A metabolism. It's likely heavily linked in some of the NADPH heavy reactions in the cell. It can help to decouple that. So knocking out doesn't help, but overexpressing can help to shuttle around that NADP and then increase tolerance. This transcription category is also very interesting. It's generally pointing to, in here mostly, medium global transcription factors, kind of pointing out that maybe knocking out global regulators isn't actually helpful because it's coordinating a complex response, which sometimes runs counter to common approach to knocking out regulators. If we jump into another cluster here, well, we see the opposite pattern. So in this cluster of 185 genes, what we're seeing is that the knockout actually helps, but the weak promoter or any other promoter generally hurts. So we call these tolerance susceptible genes, genes that knocking them out helps, but overexpressing them makes you more susceptible to furfural toxicity. And a gene that falls into this category, for example, makes perfect sense, is YQHC. In this case, it's actually really interesting. This is actually a local transcription factor. So I just told you that the global or medium scale transcription factors generally fall into this pattern. It's really interesting to see a local transcription factor here, YQHC, which specifically regulates that YQHD gene that we talked about. Knocking that out actually helps, and that makes perfect sense because this is an active regulator of YQHD. And so knocking it out is similar to knocking out YQHD. And so then we also see that knocking out things like auxiliary carbohydrate that are all genes that the carbon source is not in the media would help, and predictably knocking out some of the NADH machinery would not help, right, because you're trying to increase that pool of NADPH. If we look at the other two clusters, it's kind of what you'd expect. You see that you have the tolerance conferring, which is down and up, tolerance susceptible, which is up and then down, and then tolerance sensitive, meaning that any of these 265 genes, pretty much no matter what you do, dysregulation of them in any way seems to help with tolerance. This final cluster showing that in these 318 genes, anything that you really do to them generally hurts your overall tolerance. And so, in sum total, in the study, we can directly recapitulate 7 out of 8 of these heavily validated furfural tolerance genetic perturbations. So, everything I'm showing here, and the key aspect of this mechanism is heavily validated by the excellent work of the Ingram group. We recapitulate readily in the study. We identified thousands of genotype-phenotype connections that can be readily mined to increase or decrease a strain's tolerance to furfural. And we further show how clusters of hundreds of these genes fall into coordinated modules that can be used to select targets for predictable responses. And so with that, thinking about what's going to happen going forward, so in a forward engineering application for strain tolerance, what you might do is you might first take the top hits from all around the genome. And really, if you're looking at the y-axis, it's performance of your strain over time, whatever characteristic might be, across multiple runs of editing. You might first run this workflow, identify all these different hits. They're going to help improve your tolerance. And then you're going to recombine these combinatorially or one at a time in multiple rounds, climb up this fitness landscape, and get an ever more tolerant and more productive strain. And so at Inscripta, kind of what's coming next. So I talked to you about single edit data, making one edit at a time, thousands of those around the genome, 25,000. The next steps for releasing on the Onyx instrument will be, after the single edit kit, will be combinatorial editing kits. So here's some early data. Again, this is the percent cell fraction, percent edited cells. Where we're getting up to 2, 3, or even sometimes 4 edits per cell across E. coli and yeast. And with that, just to recap, so Onyx Platform supports all aspects of the DGTL cycle. You first design your edits in this easy-to-use web portal. You can make a genome-wide promoter libraries, knockout libraries, saturation of the genesis, whatever library you'd like to make. But then at the push of the button, you're creating them on the instrument. We ship you all the reagents and consumables. You then test this. We also offer genotyping kits and easy-to-use analysis software that lets you upload the results from your NGS back into our system. And finally, learn to help complete the cycle, the often software and solutions to help complete the full DGTL. And again, with that, just to recap, right, we're going to generate, make this output file, have this mixed population, make these engineered cells. It works on coli and yeast. I showed mostly coli data today. Current first offering is 1 edit per cell, searching solutions and substitutions. The number of designs is 10,000 in coli, 6,000 in yeast. On average, 30% will be edited edited cells. About 3 to 4 weeks manufacturing time after you hit your design in the design portal, and about 1 to 3 days on the instrument. And so in conclusion, creation of libraries with up to 10,000 diverse genotypes has never been easier. So a single push of the button on Onyx platform can achieve what may have taken many months or years in the past. Genome-scale libraries that systematically alter every gene in the genome are incredibly useful to discover new targets for any application. Hopefully we've convinced you here that through just a handful of simple selection experiments and cost-effective NGS sequencing of just those barcodes, we can generate tremendous data that really confirms all this great biology and introduces new things. And really, there's one thing that gets us excited thinking about this whole world of new and exciting biology to discover now that we can write genotypes at scale. Instead of only rereading the ones natural evolution provided us. Omics did this fantastic, but we're limited by the genetic diversity that we can study. And being able to create new genotypes to study promises to open up a new era for the bioeconomy. And again, in just really a few weeks on the bench, we were able to recapitulate years of work in stress tolerance and also identify hundreds of new targets for strain improvement. And thank you so much for listening. And with that, I will take any questions.
All right, great. Thank you so much. That was excellent. We did have some questions come in. We will get to those right away. If you still have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. So Stephen, if you're ready, we will get to our first question, which is, are you aiming at multiple edits per cell soon?
Yes, absolutely. So I know I have one slide here at the end. So you can still see this. Slide number 35, we talk about future offerings in Inscripta, which is multiplex common cell editing. That'll be coming soon after the release of our single edit kit. So shown here, we're showing that you can do multiple edits per cell at a high cell fraction, percent edited cells of 1, 2, 3, even up to 4 in some cases for Cerevisia and in coli.
Okay, great. Next question is whether you've considered the application of this technology on other eukaryotic systems, for example, on cancer cells or viral genomes.
Yeah, absolutely. So we do obviously just talking full work in yeast, but Inscripta is actively working to build tools for mammalian cell biologists as well. And that will be coming in the future.
Okay, great. Next question. Since the efficiency of editing is not 100%, how will you enrich or identify the edited population?
Great question, great question. Right, so as I mentioned, the efficiency is 100%. So in our case, there's 1 million cells, 250,000 of them. Let's see if I can do this. Going back to this setup, if there's 1 million cells, 250,000 of them will be edited. So there'll be 750,000 cells that won't be edited. In a pooled selection, the edited cells that comparisons can manage will outcompete the unedited cells. And so from a control condition versus the challenge condition for all organisms, direction for all, you will see that the strains that outcompeted the others, just like in evolution, will have a stronger representation in the population. And the sequencing of those barcodes in the comparison will show you the ones that are enriched.
Okay, excellent. Next question. Do you ever see evidence of sigma 70 saturation or depletion after inserting so many strong promoters?
Great question. That's yeah, very interesting. So in our case, for this current setup, we are inserting one edit per cell. So just to go maybe back to right here, but we're only having one edit per cell. In the case where we would have multiple, you might imagine that multiple strong promoters would be sucking up a lot of that sigma 70 and causing the growth effect. However, there even could be the case, just pointing out that at certain promoters in the E. coli genome that are in very active regions, perhaps they could consume a lot of sigma 70 and deplete. It's very interesting. We haven't looked at that in detail, but thanks for the question.
Okay. Next, how will this good result extrapolate to other bacterial species, almost for the most conservative genes?
So in terms of other species, again, the Inscripta platform is set for E. coli and yeast. And so we do support custom strains of E. coli and yeast. So if you have a production strain, that's something that should be compatible with the platform. In terms of other organisms, that would fall under future work.
Okay. Next, you showed a strong enrichment pattern for edits near the origin of replication, but it seemed to be in only one direction. Why do you think it might have been non-uniform?
Yeah, that's a great question. So let's pull that back up here. So in this case, we're seeing this really strong enrichment of knockouts from about 3 to 3.9 megabases. And the origin of replication is right around 3.9. We don't know. Again, it's one of those cases where when you're making these thousands of genomic edits, looking at this for the first time, you find new biology. A lot of the ribosomal genes do show up from 3 to 3.9 megabases. As I mentioned, that's an area where under log phase growth, the E. coli genome has multiple replication forks, and there's higher copy number genes that are more important for metabolism than essential. That is our working hypothesis for why it might be asymmetric on that side.
Okay, excellent. And we have time for one more. You showed data for making 25,000 edits. Did you actually make all 25,000? And if so, how did you characterize that? Right.
Yes. So all 25,000 edits are made. And as we mentioned, we often get up to 99% coverage, meaning that they all should have been in the population. We also showed, however, that in this case, we had 25% fraction edited cells. What we know and what we see from our data coming back is that at the very least, many thousands of those edits went in. And maybe the strongest way to look at that and think about it is when we do the GMAP clustering, we have over 990 genes where we're seeing strong responses across multiple different edit types. So, we know that many tens of thousands of edits.
Okay, excellent, great. Thank you so much. Before we go, I'd like to thank our speaker one more time and thank the audience for joining us today. We'd also like to thank LabRoots and our sponsor Inscripta for underwriting today's educational webcast. The questions that we did not have time for and those that are submitted during the on-demand period will be addressed by the speaker using the contact information you provided at the time of your registration. This webcast can be viewed on demand. LabRoots will alert you by email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. So with that, I thank you again for joining and hope you have a great day.